the day today. So just very quickly, I'm Jessica Wilson. I'm the chair of the AEESP Student and Postdoc Services Committee. We're really excited for our third Future Faculty Seminar Series. The slide that you see right now is just a way to continue to connect with AESP on social media. This was provided by the Internet Resources Committee. So I'll put this up at the very end in case you want to scan it and connect with AESP. And now I will turn it over to our two co-hosts for today's seminar, Nicole Rocky and Lauren Stadler.
So it's my pleasure to introduce our first speaker, Dr. Seju Kang. If you want to start sharing your screen, that would be great. Seju is a postdoctoral researcher in the Department of Environmental Microbiology at AEVAG. Before that, he earned his PhD from Virginia Tech and his master's from Seoul National University. So we have about a 25-minute talk, and then we'll have an opportunity to ask questions. And if everyone could please mute themselves while Seju is speaking. Okay, so I'll hand it over to you now.
Thank you. Thank you for the nice introduction, and thank you for the invitation to this seminar. So I'm pleased to share my research job and research journey and research plan for this today. The title is Transforming Environmental Health Surveillance and Epidemiology for an Equitable and Affordable Future. So a little bit of myself, as Lauren described, I did my bachelor and master in civil and environmental engineering at Seoul National University, South Korea. And I moved to US to do my PhD in civil engineering at Virginia Tech. And up until now, I've been doing my postdoctoral training in environmental microbiology at AEVAG in Switzerland. And my research interests span environmental monitoring, infectious disease, public health, water, sanitation, and hygiene. And if you think about the modern era, there has been a consistent occurrence of major epidemics from respiratory viruses or waterborne viruses, waterborne pathogen, or even like vector-borne viruses, vector-borne pathogen. There are different types of diseases are spreading across the globe in different locations. The most recent one is the Mpox in Democratic Republic of Congo. And yeah, if you look at the history of these epidemics, you can kind of expect that there will be more to come in the near future, and we should get better prepared to fight against infectious disease spread. But there's no perfect one solution to do that. There's a collective effort should be required in the multi-sectors with the best practices. For example, vaccine development or case management at the healthcare facilities, or governance leadership is important for international cooperation. But the surveillance set the groundwork for all these practices, so provide more information to understand disease spread. So what is surveillance? Surveillance to collect information, what type of the disease, when and where they're spreading. And that this kind of information helps us to set up priorities and develop targeted interventions. And the main focal point of surveillance usually is focus on the clinical sample, which means people get infected by disease, they go to hospital and they get diagnosis. But there's a still limitation of the clinical surveillance. There's an asymptomatic patient doesn't go to hospital, and some of the location has a limited access to health facilities. And there are some under-reporting issue for specific disease, for example, the cholera. And the wastewater-based surveillance has been acknowledged as a complementing tool towards clinical surveillance. In principle, to keep track of the level of pathogen in the wastewater influent that covers all the soil line. And then this information can be traced back to infectious disease spread at the community level. And then my research topic is more focused on this wastewater-based surveillance. In this talk, I want to touch on the current status of this wastewater-based surveillance in the example of my work in Switzerland. And I want to suggest a future direction of this work. And I want to suggest how we make this system more equitable and affordable. Let's start with the current state of the wastewater-based surveillance. In Swiss at AEVAG in our institute, we've been established national scale wastewater-based surveillance. We've been collecting composite wastewater influence from multiple wastewater treatment plants across Switzerland. We process them and extract the nucleic acid. And we implemented multiplex digital PCR assay for quantifying multiple respiratory viruses, including SARS-CoV-2 and influenza A and B and RSV. And this data has been made in public so that people can see there's a different dynamics of the different disease through the wastewater data. And not only keep track of dynamics of different respiratory viruses, we try to make more in-depth information from the wastewater, which is we want to understand how this virus evolve over time. So my interest was in human influenza A viruses, so specifically H1N1 and H3N2 types. This virus has an A-segmented RNA genome. So we try to achieve amplicon sequencing for these two targets. So we have a subset of the extract we collected in the Switzerland. From the 4 wastewater treatment plant. As you can see on the right figure, there's a seasonality of this influenza A in the wastewater. This is estimated by the digital PCR. In the winter season, there's a high load of influenza A was found in the wastewater. And we selected some of this influenza A positive sample. And we developed influenza A virus primer panel that targeting 3 of the interest RNA region, which is HA, NA, and N. And we use a nanopore sequencing platform to get the high coverage sequencing data from the wastewater. And then we implemented a bioinformatic pipeline called VPipe for further analysis. Some of the key findings from our wastewater data that we were able to estimate a relative abundance of the clade of H1N1 from the wastewater. And this is the wastewater data from Geneva. As you can see, there's 2 main clades was found in the wastewater, color-coded by light blue and dark blue. And this abundance changed over time. And this relative abundance is quite in line with the clinical trend, so that we can say the wastewater sequence data could complement the clinical data. And another finding is that we also identified multiple mutations from the wastewater. There are multiple mutations we were found in the graph, but more importantly, there's a key mutation we found called S247N mutation. We found one of the samples from Geneva. This is known to be associated with a mild reduction in the Tamiflu efficacy. So this is a kind of sum up of the current status of the wastewater waste surveillance. In the example of Swiss, we established streamlined logistics for sample collection and processing. We deployed quite robust assay towards wastewater, which is multiplex digital PCR assay. We also achieved great sequencing capacity, including SARS-CoV-2 and influenza and other viruses as well. And then I just want to propose my research program as a future faculty and question me, what is the future direction of the wastewater surveillance? And there will be many options. People will have different opinions. And there's one direction that I have motivates me most was the climate change. And the climate-driven infectious disease dynamics is happening across the globe. If you think about the modern era, there are a lot of travel, trade, and climate change that also influence a lot of vector-borne disease migration. And think about the dengue, chikungunya, Zika, malaria, like a mosquito transmitted disease. If you look at the figure on the right, people expect because of climate change, there's a vector migrate in the northern area. So a lot of these vector-borne virus disease could be more often found in the northern area as we move forward. So in this context, I want to develop more climate responsive wastewater-based surveillance on the shifting environmental conditions. First of all, I want to develop the panel that we can target more climate-sensitive pathogens. Also, I want to study more about the wastewater signal impacted by these environmental conditions, for example, temperature, precipitation. even now the extreme weather data like heat wave and flooding could make our wastewater signal interpretation more complicated. So that's something, the direction I want to move forward this system. And the other direction I want to go is I try to make this system more equitable. If you think about the wastewater surveillance that I showed in the figure, many people think that sanitation has just one type, but there are many different categories of the sanitation in the different places. Sanitation is defined as how human waste is disposed and treated, but not all this human waste is treated by the sewer line and centralized treatment plant. So different sanitation conditions exist. These are like a more broad, general 3 categories with different convenience and cost. If you on the right upper figure, this is like a sewer line, conventional centralized treatment plant. But there's a more small-scale decentralized treatment plant also exists in some area. And then also other people, also many people are rely on the on-site sanitation, also known as non-source sanitation. Many people use this sanitation in some area. And then the 2022 WHO reported nearly half of world population is served by this non-source sanitation. And I've been conducting project funded by AEESP. We try to transfer the framework of the wastewater-based surveillance in the urban non-source area. The project is based on Uganda, partnering with local partners listed here. So we try to transfer this wastewater surveillance is non-urban area. If you think about the difference of sanitation between sewer and non-sewer sanitation, in the perspective of the surveillance, in sewer sanitation, there's only one sampling point, which is wastewater influent that converts all the human stream through the sewer line. But meanwhile, in non-sewer sanitation, there are a lot of this, scattered on-site containments, and there's a potential pathway of the pathogen flew from the on-site containment through the environment. So to our best knowledge, there's no strategic sampling plan to do this inbound surveillance in this non-sewer sanitation. And this project is based in Kampala city of Uganda, Kampala. This is a map of the Kampala city with 5 administrative divisions and the sewer network. As you can see in the map, only central division is covered by sewer network, which means if you monitor wastewater in front only, you will end up getting information in this sewer area. So millions population living outside of this sewer network become invisible if you implement wastewater-based surveillance. So overarching hypothesis of this project is that can drainage function as a proxy for community health monitoring. If you look at the map on the right, this is a drainage channel stream in the Kampala city. The drainage channel is quite ubiquitous, running through across the city. So it has a great potential that information can represent all the citywide information for community health. This is one example of some of the example of the drainage channel in Kampala. Some of are very natural on-line channel, and some of are very constructed and the line channel. So the infrastructure is also very point by point, and if you look at the neighborhood, socioeconomic factor also quite variable point by point. After we work with the local partners and have a expert group discussion and field validation. We were identify some of the interesting drainage points, and using hydrological model, we were able to delineate a micro catchment. So micro catchment is more like a small size of catchment that capture several tens of 100,000 population. And all these blue regions are delineated micro catchment. So if you sample from drainage point, you're able to find spatial information of this micro catchment. So we selected 10 drainage points for monitoring, and we also added 2 wastewater influent as a comparison. And this is one of the example results we found. We monitor norovirus G2 in the sample, and all the numbers are indicated here representing different location of drainage. And the Lubisi and Nachibubo indicates two wastewater influent. And as expected, the level of norovirus is higher in the wastewater influent. But still, there is some drainage has a very higher level of pathogen norovirus in the drainage. But across, if you compare across different 10 drainage points, there's a high variability exists across drainage point. But interestingly, if you normalize all these signal by PNOV, which accounts for human factor, all the signal gets quite comparable even with the wastewater influent. So if you use this signal, that implies that all the drainage point has a great potential as a proxy for common health monitoring. So we just concluded the first sampling campaign and second sampling campaign last September and October. So have more data. So we expect to conclude our hypothesis in the future. So this is the one direction I want to go in terms of wastewater-based surveillance. I want to make it more equitable across the globe with different sanitation context. And the next direction I want to go is I try to make a more affordable system. If you think about doing all this wastewater-based surveillance, you have to collect the sample, process it, analyze it. Usually it quite require intensive resources to do that. For example, digital PCR or sequencing often can be only implemented in the centralized laboratory. And not all the places has this intensive resources, and there's a lot of challenges exist over there. So that's kind of ignite my passion towards next generation of pathogen sensing. I try to develop some methods that are applicable under resource limited settings. So try to make it more system, more low cost and user friendly, doesn't require professionals to do that, and rapid and portable. And it would be nice to have a minimal sample preparation, so make it more simple, streamlined workflow. But also keep the good standard, like offering sensitive, specific, and robust detection as to conventional method as well. During my PhD, I've been using technique called SERS, Surface Enhanced Raman Spectroscopy, to develop a less resource-intensive sensing platform. And I also use the technique called loop-mediated isosomal amplification, also known as a LAMP. I did use this LAMP during my PhD and postdoc. And also, and lastly, I want to use, I did use a user-friendly deployable molecular analysis platform developed by the starter company at ETH, and use this for capacity building in other countries. Start with the source. Raman scattering is in elastic scatter light. Basically, inelastic means there's energy change when it's scatter light. A very small fraction of scatter light there's an energy change, which is reflected by the Raman shift. So, and this unique Raman shift collection of Raman shift can function as a molecular fingerprint. But the fraction of this inelastic scatter light is very small. Even though it has a great specificity, the signal is pretty low. There was a huge challenge. But in 1974, researchers found that if a target of interest situated near plasmonic nanoparticle, the signal can be significantly improved, for example, gold nanoparticle. And this phenomenon is known as a surface enhanced Raman spectroscopy. So basically, you try to put your target of interest for sensing, you put it in the near the waste gold nanoparticle or plasmonic nanomaterials. So this source signal can have achieve very sensitive sensing platform. The main benefit of source is a low cost and sensitive, and because you use a laser-based detection method, it's non-invasive and easy to use. It also has a function of a portable device and a minimal sample preparation. And during my PhD, I have been developed multiple cell source-based environmental sensing platform. So fabricate multiple plasmonic nanoparticle for the different goals. For example, I made a gold nanoparticle and combined with a magnetic nanoparticle to get more target separation. Or I fabricated gold nanostructure substrate to enable more microenvironment pH sensing. and also try to aggregate cold nanoparticle or make a porous supraparticle for concentrate the target for better sensitivity. And for the biological targets like a bacteria or virus, I try to functionalize cold nanoparticle with the lectin or combine with the culture-based method. So different goals require different strategy or workflow for source-based sensing platform. The next technique I use is a loop-mediated isothermal amplification. So this is a molecular method that amplifies target of interest, similar to PCR. But unlike the PCR, they use a 6-primer set with 3 different functions. So the benefit of the lamp is that it's amplify target of interest very rapidly, and the isothermal condition, which is constant temperature, so it doesn't require any thermal cycling like PCR. Using this technique, I've been developed a duplex lamp assay for diarrhegenic E. coli in the wastewater. And further, we try to make this readout a more point of care setting. So we use a lateral flow, so it has an easy to use and quick for diagnostic tool. And also, I've been developing duplex lamp assay as well for Vibrio cholera in the wastewater. And during my PhD also, I use this lamp and combine the nanopore sequencing, is also known as LAMPORE. So basically, you use a nanopore sequencing as a readout of the lamp amplification. The benefit of this combination is that you can insert unique barcodes, multiple barcodes during the lamp step and the library preparation step during the sequencing. And then this combination of barcodes can make this assay very highly multiplex. So in this platform, we were able to analyze 96 samples of the wastewater extract for SARS-CoV-2 in just one sequencing. Not only high multiplexity, you can see the sequencing data. So we were able to identify the Q mutation for SARS-CoV-2. The last technique I use, this is a rapid and easy to use molecular tool for cholera surveillance. This project is funded by Swiss Humanitarian Aid. So I work with the ETH spin-off company. So this company invented the device, all-in-one device that can automate extraction and PCR. The advantage of this tool is that they have a customized part for the target, assay for for specific target, and this assay can be ready in the preloaded reagent in the pod, so it doesn't require any cold chain transport. So you can ship this assay to anywhere without cold chain transport. And the other benefit is that they developed a unique material that facilitate PCR reaction, facilitate a heat transfer. So they shorten this PCR reaction time within the 30 minutes. So this device is quite innovative, because if you prepare sample, put it in, push the button, all this extraction and PCR can happen in 1 hour. So we use device, we do the field test and the training program in Lusaka, Zambia in last April. Basically, I was in charge of shipping this machine, and then we met the technician and Lusaka Water Supply and Sewer Corporation people and tried to train them what's the PCR, what's the principle, why it's important, and how to use device, what kind of information we can get from this. And this is done in the last April. So with that, I just want to conclude my talk again. To summarize, I was working on the wastewater surveillance. I touched on what's the current state of the wastewater surveillance. And then one of my future direction I really like to try is to try to make a more climate responsive system. And more further, I want to make this wastewater-based surveillance system more equitable, especially in the non-sewer area, and also want to make it affordable so that all the tools that we use can be applicable in resource-limited settings. With that, I just want to acknowledge and thank all of my colleagues and peers that I've involved in my journey of PhD and postdoc. Thank you for your attention. I'm happy to take any questions.
Thank you so much for that great talk. Okay, so now we'll open it up for questions. I believe you can raise your hand or you can just unmute yourself if I can't see your raised hand. Okay, Nicole has a question.
Yeah, I'll go ahead and I have a maybe naive question about SIRS.
Yeah.
I'm curious, and you might have said this, but is your like vision for the SIRS work to be non-targeted, so it could hit like the signature would be multiple different types of microbes, or is it like you'd have to specifically target an individual microbe? Or could you talk more about what you're thinking?
Yeah, I mean, because the third spectrum has a unique to the biomolecule, so it can be non-targeted strategy. But if you want to really have a one target of interest, you can design you can fabricate your nanomaterial, like functionalize their surface. For example, like a functionalized antibody to specific the bacteria, or you can functionalize surface by DNA aptamer to target DNA sequences. So it can be targeted or non-targeted as well, depending on what you want.
Great. I have a question.
Yeah.
I'm curious about for your drainage study where you compared the different drainage sites to the downstream treatment plants. It was really interesting to see the normalized data for a very abundant pathogen that's kind of present in most samples. Did you look at any rare pathogens, and did you have any instances where maybe you saw things only present in the drainage and not at the downstream site, or was that not part of the study?
We've been monitoring multiple enteric viruses and respiratory viruses. So if you, for the enteric viruses like a norovirus, rotavirus, there's a constant detected in the drainage system. But if you, we monitor also respiratory viruses, like we designed the 6+ assay for SARS-CoV-2, influenza A and B RSV. So we did detect some of the influenza A signal in the drainage as well. So yeah.
But do you ever see it like in the drainage and not in the downstream treatment plant? Like, are you afforded any additional sensitivity by going upstream?
There's a, for example, influenza A, there's a few positive samples in the drainage, but there were no influenza A in the wastewater influent. So yeah, I think drainage can capture some pathogen that cannot be captured by wastewater influent. Yeah, we see some differences.
Great. Well, I have another question if no one else has another. Okay, so my question, I really liked your suite of technologies that you presented kind of for on-site or lower resource settings. And I guess maybe you could speak a little bit about like where you see each of those having a unique niche in terms of application. And then maybe a more technical question is about your LAMP LAMP followed by sequencing. Are there unique challenges associated with designing those primer sets for the multiplexed LAMP and sequencing workflow?
Yeah. In the specific case of LAMP, like I kind of struggle to make it multiplex because it involves multiple primers. Like a PCR uses forward and reverse primer and probe, but the LAMP there has a 6 primer and the concentration are usually higher than PCR. So we found a lot of non-specific applications when you make it multiplex. And yeah, this is, I think LAMP is quite known for high rate of false positive. So I think that's one of the key challenges compared to PCR.
That's helpful.
Yeah.
Okay, well, if there are no additional questions, let's thank our first speaker one more time, and I will hand it over to Nicole, who will introduce our next speaker.
Great, thank you all. So I'm excited to introduce today Dr. Mary Kate Lane, who did her PhD at Yale University in civil and environmental engineering, and then is now at MIT in a consortium, the Climate and Sustainability Consortium, as an impact fellow as a postdoc. So we are excited to hear from you, and I'll go ahead and you can take it away.
Thank you so much. Yeah, so I just wanted to start with a brief introduction. So I am Mary Kate Mitchell-Lane, and I'm a chemical engineer by training, receiving my bachelor's from Michigan Tech. And then I went on to earn my PhD in chemical and environmental engineering with Professor Julie Zimmerman at Yale. And while in undergrad, I had the unique opportunity to do several internships and co-ops in the energy industry. So I worked at oil refineries in Ohio and in Texas. I also worked for Michigan's largest natural gas and electricity utility, primarily on their natural gas pipelines. Then I also worked for a small energy storage device startup in Boston. And these were all very formative in my early years and really informed how I think about the energy industry and why I wanted to do environmental engineering for my PhD. I'm currently a postdoc researcher at MIT in civil and environmental engineering, working with Dr. Desiree Plata. And then I also have this co-appointment with the MIT Climate and Sustainability Consortium. So as an impact fellow with the MIT Climate and Sustainability Consortium, I foster collaborations between MIT and our member companies, so large industrial partners, to accelerate the implementation of climate and sustainability solutions. But today I'll be sharing my research vision for defossilizing the chemical industry through design and deployment of an agile and sustainable bioeconomy. So I love starting this talk with a vision of innovation as waves that have pushed humanity forward technologically throughout history. And I want to highlight how chemistry and engineering have been the drivers of these waves that allow us to live in the world we do today with all of our modern technologies and amenities. So these waves of innovation reshaped human experience that have consequently led to a host of environmental problems, including the current climate crisis. And these environmental problems stem from the design of current systems that rely on fossil carbon for energy, chemicals, and materials. So with this reality, we need to radically redesign society's relationship to carbon from a system that is linear, extractive, and polluting, taking this long-term stored fossil carbon directly to the atmosphere when used for fuels, to a new system that's circular, regenerative, and sustainable, while still meeting the needs of society. So while road transportation can be electrified and large parts of the energy sector can expand renewable energy to decarbonize, eliminating carbon entirely isn't feasible for liquid fuels, chemicals, and materials where carbon remains essential to the function it provides in society. So decarbonization may not accurately reflect the chemical industry's needs. Instead, we can defossilize by replacing fossil-derived carbon with renewable sources of carbon to meet global needs. So one way to defossilize is to use biomass as a renewable feedstock, as the biomass carbon cycle draws down carbon from the atmosphere in a circular and regenerative nature. And biomass systems also have the potential to be sustainable when implemented responsibly. So going back to our waves of innovation, scholars contend that humanity has entered a sixth wave of innovation through new technologies like artificial intelligence, that is paired with a new perspective focused on sustainability through the rise of renewable energy and clean tech. So my proposed research sits at the crest of this wave and aims to combine expertise from several nodes of innovation to design and launch a fossil carbon-free future. I will also focus to prepare the next generation of chemists and engineers for careers in this sixth wave through empowering them with the technical knowledge to make an impact, while emphasizing sustainability, green chemistry, and systems thinking. So as chemists and engineers and material scientists, as a field, we have completed substantial work to develop processes and techniques to transform renewable biomass into biofuels, biochemicals, and biomaterials. But the complex nascent bioeconomy requires robust analysis and novel process innovations to be implemented efficiently, economically, and sustainably. So my research seeks to fill the gaps that exist in both design and deployment of the bioeconomy through first designing chemical reactions, extractions, and processes to produce high-value chemicals and products in integrated biorefinery schemes. And then also looking to fully utilize biomass for food, fuel, chemicals, materials, carbon storage, and to maximize the climate benefit at economically competitive price points. And then finally, deploying machine learning and artificial intelligence, these tools of the sixth wave, to design, optimize, and implement biomass-based chemical production systems at scale. So overall, I aim for my future research to have this impact from design of processes to deployment in the broader bioeconomy. And today I'm going to use my PhD research focused on algae biofuels and the integrated biorefinery to provide context on how I see a possible trajectory for the bioeconomy to power the chemical industry and how I'd like to make an impact in my future research. So first, to be upfront, I'd like to share a cautionary tale of algae biofuels that you may know. In the mid-2000s to the early 2010s, biofuels derived from algae were going to save the world from global warming and were the next big thing for venture capital to funnel their money into after the dot-com bust. So this article from 2007 exemplifies the promise of algae biofuels. If the US put 15 million acres of desert into algae production, we could produce enough volume of liquid fuels to get us off the Middle East oil addiction and give Iowa back to the songbirds. I think this is a beautiful vision, but here we are in 2025, and we don't have bountiful algae biofuels, and Iowa is still mainly corn and soybeans. So what happens? One of the main downfalls of algae biofuels were simply the economics and competing on price point with fossil fuels, and the singular focus of fuel as the only product. In fact, when you consider a conventional oil refinery, they produce much more than just fuels. A barrel of crude oil goes to fuels, but also produces a range of bulk chemicals, specialty chemicals, and pharmaceutical precursors. And looking at the breakdown of this barrel of oil, we can see that by volume, fuel products are the largest, with chemical products only accounting for about 16% by volume. But this 16% by volume accounts for almost half of the value derived from a barrel of oil. So conventional oil refineries produce both high volume and low value fuels, and then low volume, high value chemical products to fully utilize the entire barrel and maximize profit. So we can take this idea, moving from a conventional oil refinery to an integrated biorefinery, by swapping out fossil crude oil with renewable sources of biomass. And then the integrated biorefinery fully utilizes renewable biomass feedstocks through extraction, separations, and reactions, and ideally with green chemistry methods, to produce many of the same products that are currently sourced from petroleum feedstocks, and produce multiple products at varying price points. And then in addition, since we are not limited to the chemical structures in fossil crude oil, we have the opportunity to obtain additional products like nutraceuticals that are naturally derived compounds with health or medicinal benefits. So my PhD research focused on developing integrated biorefinery schemes with algae biofeedstocks. And during this work, I produced 2 publications and I mentored 5 undergraduates in lab. And then 4 of them contributed significantly to be co-authors for this research as well. So here is Rachel and Mary Ben in front of our Center for Green Chemistry and Green Engineering at Yale. And so with the algae biomass, I specifically looked at Phaeodactylum tricornitum microalgae because this algae naturally produces multiple high-value products. So Phaeodactylum tricornitum microalgae, or PHAO, contains both a triglyceride and a carotenoid fraction. And so the triglycerides include tags for biodiesel, and the biodiesel can be created by breaking off these fatty acid chains from the glycerol backbone here. And some of these fatty acid chains are actually worth more as nutraceuticals, like eicosapentaenoic acid, or EPA, and dodecahexaenoic acid, or DHA. So these are both omega-3 fatty acids that you can take as a supplement for your health. And then the carotenoid fraction are colorful pigments with this central chromophore, which absorbs light at a particular wavelength and emits color as a result. So some of the ones in PHAO include this bright red-orange fucoxanthin, green chlorophyll, and then yellow-brown beta-carotene and zeaxanthin. And several of these are nutraceuticals. And I'm specifically going to look at triglycerides for biodiesel and fucoxanthin today. Fucoxanthin is a high-value nutraceutical, and it's an emerging antioxidant with benefits for cancer, obesity, and liver disease. So the goal of the integrated biorefinery is to maximize the value of biomass feedstocks by efficiently utilizing all components and creating multiple product streams at varying price points. So for microalgae, I can target the low value fuels for the triglycerides, and then I can also target high value nutraceutical fucoxanthin. And so here we can see that the fuels are valued about $4 to $5 per gallon, or about $1 to $2 per kilogram. And then fucoxanthin is $40,000 to $80,000 per kilogram. So a very small amount can be worth a lot. Additionally, I use green solvents, supercritical carbon dioxide and ethanol for tunable extractions to produce these multiple products. So instead of using volatility to separate compounds like in distillation columns, this method uses polarity and changing pressure to target different fractions. And the two main goals of this research were to identify conditions to selectively target these multiple products, and then to design selective sequential extraction schemes to produce high purity products at high recoveries. So I used supercritical carbon dioxide in my method because it's green, as in non-toxic, it's in every breath we exhale, and because it's adaptable and tunable. When you look at CO2, it is a linear molecule, but it is actually quadrupolar, meaning that it has intermolecular polarity as the molecules are pushed closer together. So CO2 is polarizable with the ability to act as a more polar solvent at increasing densities. In the supercritical state, above 31 degrees Celsius and 7.4 megapascals for carbon dioxide, small changes in temperature or pressure result in large changes in density. So the density, and therefore the polarity, can be tuned by setting the temperature and pressure in the supercritical range. And polarity or polarizability, the ability of a molecule to become polar, can be quantitatively measured through what's called a Camelot-Taft parameter called pi star. So we can use pi star as a measure of solvent polarity, where on a relative scale, lower values are more non-polar and higher values are more polar conditions. So here we can see for increasing density in CO2, it correlates very well with increasing pi star as well. And so we can set polarity in supercritical fluid extraction systems by either increasing the system pressure at a constant temperature, and in this case, I'm looking at conditions of 13 megapascals, 30, and 50 megapascals. You could also add in a polar co-solvent like ethanol, which is also considered a green solvent because of its low toxicity. And in this case, I'm looking at 5%, 10%, 40%, and 100% ethanol to look at a range of polarities in my extractions. And so in my experimental setup, I kept the temperature constant at 50 degrees Celsius in a heated oven. Inside a pressurized reaction vessel is our sample is loaded. And in this case, I'm looking at just pure fucoxanthin because I wanted to see without the effect of the algae that it's being pulled out of, what was happening. And then also from our algae, the fucoxanthin being extracted directly from the biomass. So these two were loaded into our vessel, and then CO2 was pressurized and pumped through the vessel and then out the exit valve. When it was just pure CO2, the CO2 rapidly depressurizes and kind of sprays our product that it had solubilized from the bed of algae biomass into our collection vial. When I used ethanol, I pumped ethanol in and it mixed with the CO2 at a mixing tea, and again was collected in the vial. So for these results, I first looked at fucoxanthin as a pure standard. And so we have our results in systems that are just supercritical carbon dioxide and the ones that include ethanol as their co-solvent in the X's. So looking at these results, first we confirmed that when you increase the pressure in CO2 and increasing polarity, you do get an increase of fucoxanthin recovery. And then with including increasing amounts of ethanol, we also see the same trend. And then with both of these results together, we can see that at increasing polarity, we do increase fucoxanthin recovery. And so there's two important findings in these results. And the first is that we can achieve 100% recovery of fucoxanthin around polarities of 0.2. And so this is only with about 40% ethanol. So we actually don't need to be at extremely high polarities or full ethanol systems to achieve full recovery. So this would save on solvent needs when designing our sequential extractions. And then the second point is that we also reach a limit of recovery in pure CO2 systems. So I'm pointing here at our highest pressure that we tried. And this is an important finding because it illustrates that even when fucoxanthin is entirely accessible, because it's loaded as a pure standard, supercritical carbon dioxide is not able to recover all of the fucoxanthin. So here at the highest pressure, it was about 30% recovery at 50 MPa or 7250 PSI. And if we were to extrapolate the polarity in a pure CO2 system to what pressure that we would need to be at, it actually exceeds 10,000 PSI. And so that was above the operating limit for our pressure vessel and would likely have implications for the engineering of pressure vessels as they get more expensive as you need to be rated to extremely high pressures. And so we also looked at extractions with the same conditions, but now loading in our algae and pulling fucoxanthin from the algae. And here we saw that there was a definite effect of pulling fucoxanthin from the algae than when it was fully accessible. And so this illustrates that perhaps there are interventions in algae pretreatment or algae types that could boost recoveries to these theoretical maximums established by the extractions of fucoxanthin standard, since it wasn't as freely as accessible when it was being pulled from the algae. But it does still follow this trend of increasing polarity and increasing recovery. Another interesting finding from this first fundamental study is that supercritical carbon dioxide systems showed evidence of selectivity over mixed ethanol and CO2 systems. If we look at these 2 points, which are similar polarities, and the 2 samples that I collected out of the end of the extractor, you can see that one is this bright green color and one is this bright orange. You can actually see this in the algae itself before and after. So this is what the color looked like before. It's always fun when chemistry is colorful. It's kind of this brownish algae. And then after the CO2 extraction, the orange pigment is pulled out, the fucoxanthin, and it leaves this bright green behind. Where after extractions with CO2 and ethanol, it pulls out some of the green and the orange at the same time in this sample. So we can kind of see that when we track the chlorophyll of the algae quantitatively, it matches what our visual observations were. And so in these 2 that are similar polarities, much more chlorophyll comes out when ethanol is present than in pure CO2 systems. So pure CO2 systems are selectively leaving the chlorophyll behind in the algae and still extracting fucoxanthin. And you can see that here with the chlorophyll results. And so overall, this first fundamental study can now be used to identify selective supercritical fluid extraction conditions. And I was able to target different fractions with low polarity, mid polarity, and high polarity conditions. So if we look at this polarity down here, this -0.2, it does not pull out any of the carotenoids, the fucoxanthin or the chlorophyll. So we can target triglycerides in this step as established in previous research. And then if we wanted to target fucoxanthin selectively over chlorophyll, we can increase pressure to a mid-polarity step in a pure CO2 system. And then finally, if we wanted to target a high yield of all carotenoids, we can increase the polarity further to maximize that recovery. And so then overall, I tested 3 different sequence of polarity steps to look at how they influence recovery and purity of products. So in the first scheme, I loaded the microalgae powder into my extractor and then increased the pressure and the polarity sequentially for a triglyceride targeted extract, fucoxanthin targeted extract, and then a high yield carotenoid targeted extract. In these 2 schemes, I did this first pre-extraction step where I targeted the extraction of the carotenoids and then dried off the ethanol and then re-extracted using a triglyceride and fucoxanthin targeted steps in differing orders. So these were the real samples that I extracted from my samples. And looking at, just for the sake of time, the recovery of fucoxanthin in just the fucoxanthin targeted mid-polarity step, there is a definite impact of the order of steps when looking at the recovery of fucoxanthin, where we can get really high recoveries, which hadn't been seen in previous literature when we do this pre-extraction step, because it is more fully accessible. And then looking at purity, we also do see there's a trade-off of purity and recovery when we're looking at the schemes. As in this first step, we actually pull the triglycerides out with the fucoxanthin. And so why does this all matter? It all goes back again to the profitability to make an integrated biorefinery work. So the cost of algae and the sequence of extractions both impact profitability. So again, if we just focus on the mid-polarity step and the price of fucoxanthin from $40,000 to $80,000 per kilogram, and then we look at two methods of growing algae. We have a high-cost method, about $400 per kilogram of dried algae, which is about what it cost to grow it at NOAA Fisheries that I collaborated with during my PhD. And then also the most optimistic low-cost algae at about $2 per kilogram grown in these outdoor raceway extractors. We can see that for profitability, borrowing from a TEA of other supercritical CO2 extraction processes, you can actually lose money when you're paying really high cost algae. And it really matters that you have high recovery to really maximize the profit from your algae. And so again, this is with just the fucoxanthin for profitability. And if you can make your scheme as profitable with just this, then you can also extract the lower value triglycerides for biodiesel applications. And so when I think about the future of algae as a biofeedstock, it was really promised as this silver bullet that it didn't turn out to be. But instead, when we think about the complexity of bioeconomy feed systems, I see algae as more of one of 1,000 BBs that could take down climate change. And so if you have this algae biofeedstock and you wanted to make it economical through the extraction of fucoxanthin, you would definitely max out your fucoxanthin market before you would actually come close to maxing out any sort of biofuel market. But it can still displace some fossil fuels through the recovery of these products. And so with the other 1,000 BBs, each displacing a small amount of fossil fuels, is kind of how I see an actual economically possible system for biomass feedstocks. And so we really need to focus on both the high value products and full utilization. So borrowing from green chemistry, there's this E factor, which is the mass of total waste over mass of product. And we can also see in the oil refining as a very good E factor, producing very little waste. So if we're thinking about trying to defossilize and moving from oil to biomass, we also need to look at every waste as an opportunity and really look at food, fuel, chemicals, materials, and also carbon storage. Because even at the end of my extractions, I still had leftover algae biomass that could be used in durably storing the carbon containing in it. I also want to maintain the molecular structure at its highest utility through the design of these systems. When looking at scale now of what we're actually trying to displace for fossil fuels, the Department of Energy in the United States has this US billion-ton report that assesses how much demand for liquid fuels remains after electrification of a lot of on-road transportation. And they see that primarily for aviation and for some other applications, we would still need 7 billion gallons of liquid fuels. And this would require about 1.3 billion tons of biomass in 2050. And in their report, they are able to get to this 1.3 billion tons through several different types of feedstocks. And one that they really emphasize is agricultural residues. And so my postdoctoral research actually aims to maximize the climate benefit of agricultural residues through both fuel and carbon storage. And I'm doing this through pyrolysis experiments in lab and also geospatially resolved life cycle analyses. So I'll talk a little bit about this. So this project called End Use of Agricultural Waste for Fuel or Carbon Storage is really motivated by the fact that a lot of the current crop residues are either intentionally burned, which contributes to air quality and human health impacts, or generally just left unfueled. So this capture, or this carbon that they've captured from the atmosphere is just rapidly released back to the atmosphere. And so it can be claimed for either fuel or carbon storage applications. And I'm mentoring a PhD student and an undergraduate student for this work. And so our research questions for this project, well, we wanted to calculate the life cycle carbon impact for agricultural wastes in different end-use scenarios. We also wanted to model these wastes supplied globally with current utilizations. And this is because we wanted to only take the sustainable amount of what residues off the field. And we didn't want to change uses that were beneficial already, so for animal or other uses. And we found that about 30% of the residues left on field can be removed sustainably, and the entire fraction that is burned can be routed for climate benefit. And then we're also in the process of developing this geospatially resolved decision tool. And so in this tool, which we're calling the Ag Reuse tool, it essentially has data about the amount and type of agricultural residues globally, and then also integrates our LCA results with local carbon grid intensity, so that when you're using it for a fuel application or electricity, it can take into account the greenness of the grid and the overall results. And so we have different life cycle assessment results for each spot on the grid. And so at the end of this, where I'm hoping that my research and my future lab would be, in this sixth wave, really to meet the demand for fuels and products sustainably, and to break out of fossil crude oil chemistry restraints. And in this way, there's a potential to discover and create even better performing products when you consider the additional chemistries that are available in nature. And then I also want to prepare the next generation of chemists and engineers to be ethical and technically sound for careers in this sixth wave. And then one last thing is that I always try to also have an impact beyond teaching and research. So during my PhD, I was also I also led 2 papers that really meant a lot to me. The first was what to expect when expecting in lab. And this looks at unique risks for pregnant researchers that work in chemical laboratories, and basically lists all the things that they have potential to be exposed to, with some resources on getting more information. And then it also highlights how green chemistry can help make labs safer for everyone, and in particular pregnant researchers. And then the second paper is Green Chemistry is Just Chemistry. And this looked at the synergies between green chemistry that is trying to eliminate toxic substances and chemistry methods, and environmental justice, which typically has been overburdened with toxic substances in their communities, and how these goals align and how they can work together to promote an equitable future. So I wanted to say thank you to my affiliations and my mentors over the years and current mentors, and wanted to thank the AEESP for allowing me to share my research vision today. So thank you very much, and I'd be happy to take any questions.
Great, thank you so much for that interesting talk. We have just a couple minutes left for questions, so if you do want to raise your hand if you have any questions, feel free. All right, Liz.
Hi, thanks for your talk. It was really interesting. So did you ever consider, I know you talked about life cycle assessment in your second project. Did you ever consider that for your first project? Because, for example, the high pressure CO2 is pretty energy intensive. And so, like, is there a benchmark that you could compare your flucloxanthin, and I had to write that down, that synthesis from algae, like, to an alternative method, and compare them in terms of CO2 emissions per kilogram of flucloxanthin or something like that? Did you ever do any of that kind of analysis?
I did not, but there are some really good papers in the literature that did go through extensive life cycle analysis of algae for biofuels, and then also for nutraceuticals. So I did use one paper in my results that did a TEA and LCA. So it's this one from Journal of CO2 Utilization. There's other ones from the Zimmerman group at Yale as well that looked at algae biofuels. And I'm trying to think of the exact number off the top of my head in terms of kilograms of CO2 per functional unit. I do not have that at the moment, but I could send you the papers that I've been referencing.
It's okay. Often the CO2 mirrors the cost because it's energy and cost are correlated. So I just wondered about that, but thank you.
I'll go ahead and ask a question. I guess mine is more of thinking forward. You mentioned that there are other high value products that maybe we should be focusing on in terms of the algae biofuels. I was curious if you could speak to some of those. I like this nutraceuticals idea I hadn't really thought of before. And so, yeah, maybe other areas that you're thinking could be useful for high value products.
Yeah, so there's even a lot of like the pharmaceutical precursors that fossil fuels to currently go to. So one of them is like the active ingredient in Marilax, which can actually be derived from glycerol, which is also a byproduct of biodiesel. So it's like a waste product of a waste product that you can upgrade then to a pharmaceutical. So there's all these little opportunities, I feel like, in the bioeconomy. And so that's kind of getting to my the third point, which I didn't really elaborate on too much at this point, but really the way that we can optimize this bioeconomy is that there will be all these different types of biomass with all of these specific compounds. So there's like a lot of work going on in lignin upgrading, even like materials, biomaterials that have higher value than just like burning the biomass for fuel. So I really see fuel as the like lowest value application, but it has the highest climate benefit by defossilizing. So it's kind of like this mismatch where the economics don't really match with the climate benefit here, which also could be solved potentially by like a carbon tax or something like that. But in our current system is what I'm working with.
Great. I know we are past time. I think we technically can be on here till 1:15. So if you all need to head out, we understand. But if there are other questions, we could keep on for a few minutes.
Maybe I can ask one question. And you talked a little bit about this, or I missed it, but there's kind of a range, especially for these very high value products like fucoxanthin, where it's like, what did you say, $40,000 to $80,000 a kilogram. How much is the like purity of some of these products, especially when you are extracting them from like more waste sources, a challenge or a driver of the cost?
For fucoxanthin, since it's technically a nutraceutical, it's derived from algae.
Mm-hmm.
A lot of the pills that they sell, so there's a couple on the market. There's one called Fucovital and there's one called Thinogen. They're about 2 to 3% fucoxanthin, and then the rest is like oils in the capsule. So since it's coming from algae, it's already kind of, I guess, past the check of like impurities that you wouldn't want to have because it's already coming from something that you could just eat right now. Where I know if you were like synthesizing it from specific chemicals, then you'd maybe want to go to more pure and then dilute it down. So more pure, obviously, you could dilute it down into whatever percentage you would want it to be. But some of these products right now are already pretty low. But then that 2 to 3% is still a lot of money per capsule. Yeah.
Great. I would ask one more quick question about your work on the agricultural reuse.
Yes.
And using the residues. I guess I'm curious how you see that working in practice and what might be some of the barriers to that not currently happening and how that might shift in future to make it feasible.
Yeah, so currently right now, the residues are distributed in nature because farming is distributed in nature and agriculture is distributed in nature. But when we look at like the scale of agriculture, it actually matches pretty well to like the scale of what we need to do in terms of fuels and carbon storage. So right now, agriculture on the global scale is about 20 gigatons per year. And the latest IPCC report, they're saying that we need to have carbon dioxide removal at about like 6 to 10 gigatons a year by 2050. And of that 20 gigatons of agriculture, about 7 gigatons is our residues. And so that kind of matches pretty well. And so it really has to do with like the collection and the monetization of these residues to be beneficial. So we already move 10 gigatons of food crops around. It would just be the additional incentive of why should we also collect the residues, or how you can try to change cultural practices of on-field burning. So there is a lot of work. There's a paper I just read recently called Money Not to Burn, where they're paying farmers in India to not burn their crop residues, primarily for ecosystem services like air quality, because of the negative impacts that it has. So you can think of like similar schemes where you can pay farmers for their residues and then transform them for fuel or carbon storage, depending on what the life cycle assessment says is a better result.
Awesome. Cool. Well, I guess we are certainly past time, and we'll give people back their time. Sorry for going over there, but maybe we can all thank our speakers again for those that are still with us. Those are great talks.
Thank you so much.
Thank you.
And thank you also to our co-hosts, Nicole and Lauren. Our fourth and final seminar will be on Friday, December 5th. So please join us there if you're available. And again, these recordings will be posted to the AESP YouTube page. All right. Thank you and enjoy your Friday.
Thank
Thanks,
you.
everyone.
Bye.